What's our time?
Oh, I just stopped at the press recording.
Okay. So integrins are integrated all the way through the plasma membrane, whereas this would be a peripheral protein that's just on one side, or it could be just on the other side. Good? Other things that the extracellular matrix do: support the cell, stick it to other cells by holding on. It helps in movement and then regulation because it has receptors. Alright, we already talked about plasmodesmata and plant cells. These other three are ways that cells communicate in animal cells. So we've got, in animal cells, tight junctions, desmosomes, and gap junctions. Looking at the picture, which ones of these is most like a plasmodesmata? The gap junctions. So that's going to allow stuff to go from one cell straight into another cell. Okay, so this is good for cell communication. The cytoplasm actually can move from one to the other. Okay? Desmosomes are like those in your genes, how sometimes they have those little rivet things. Okay? So it's like binding layers together. Does that make sense? Do you know what I'm talking about? Or staples, when you staple papers together. So that, it strengthens it. So these are, this cell and this cell are tightly bound together at that location. Right here because of the desmosomes. It's like staples. And then your tight junctions make it super stuck together so that there's not going to be any leakage. So that's good for water.
They're not as strong.
I guess it depends on how much surface area they're taking up. Okay, cool? You got a chart that has all this stuff. That's good for chapter 6. Questions about chapter 6? Okay, go ahead and stop that one.